Everybody, we just talked about gene expression, and we're going to continue that conversation today. So far, we've talked about how DNA can be read into its chemical cousin, RNA. And remember, we're only copying a portion of DNA called a gene, and that makes RNA. Specifically, we're talking about mRNA, a message. And so that message is read. What happens when we read it? Well, we make a protein. So the end goal here of gene expression is to make a protein. Proteins make your traits. So for example, there's a protein for skin color called melanin. There's a protein for keratin, which determines your hair and nails. There's a protein for enzymes in your gut that help you break down food. And there's proteins for things like your immune system. There's proteins pretty much everywhere. You, as a living organism, are made of a lot of protein. So what actually is a protein? Well, it's a really complicated shape that looks like this. Where do we get it? From food like this. So you see there's a lot of meat, beans, stuff like that. Protein can also be found in like supplements. So you can get it from like this. Protein's not in just meat though. So for example, you can find it in peas. I actually take a type of protein called pea protein that I really like. You can find protein in almost all the food that you eat because most living things are made of protein, and we're breaking that down. But it's not so simple as just we become what we eat. If I ate chicken, I don't become chicken, right? Instead of becoming chicken, all I do is I break down that chicken into something, and then I turn it into human protein. So I'm taking chicken protein and I'm turning it into human protein. But I have to break it down first, right? This is a lot like having a Lego spaceship. That is an ugly spaceship. And I want to make a Lego boat. How do I go from having a Lego spaceship into having a Lego boat? Well, first I got to tear apart the Lego spaceship, right? So I take it apart and I get all these interesting blocks, right? And of course, all those blocks have different shapes and sizes. In a similar way, we do that with our food. We take something like chicken protein and we break it down and then we turn it into human protein. And so you don't become part watermelon here. She's going to break this watermelon down in her gut, not just her stomach. Keep that in mind. And she's going to turn that into human protein. And so we're going to talk all about what's in between. What are the pieces of a protein? The pieces of a protein are called amino acids. And there are 20 different varieties of it, and different foods have different amino acids. So for example, if you eat chicken, you get a lot of amino acids, but not as much actually as if you eat turkey. So it turns out turkey is a much, much better meat for you than chicken is. Consider switching to turkey. It just has more of these 20 amino acids, and that helps to build more of you, right? So different proteins need different amino acids. And let me show you what that actually looks like when you do put together a protein. So again, this is what a protein really looks like. You'll see in this video I'm going to show you that they use circles to represent proteins because it's simple. But the reality is every one of these pieces here is a different amino acid. And you can see there's a lot here. This is able to make a very complicated shape. In the same way that Legos need to be different, so do amino acids, so you can make different shapes. This particular protein will have a particular job dependent upon its shape. All right? Let's watch the video and see this all in action. We're going to watch as the RNA code becomes this protein from the food that we eat.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a
Notice that the gene is only a portion here, right? You've got all of this other stuff. Think of all of your DNA as like a cookbook, right? And then think of the chapters in that DNA as the chromosomes. So this would be a bookmark. Each chapter is a chromosome. So you would have 23 of those chapters. A page out of this is a gene. And so a gene is a lot like a page out of a cookbook. When you want to make dinner, do you make the entire cookbook? Of course you don't. You make one recipe. Your cells do the same thing. There would be no sense in reading the entire DNA sequence to make one protein like melanin. Instead, you just read one gene. And so one gene usually codes for one protein through gene expression. That, of course, has two steps. So let's watch.
Continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression.
Remember, this is how they're representing a protein here with these tiny little circles, but the reality is much more complicated. In reality, we know they really look like this. And instead of circles, we have these complicated shapes that make complicated proteins.
For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in three stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule.
All right, so question for you. If this is my, oops, If that is my DNA sequence, what will the RNA sequence be?
Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons...
So this is called an amino acid table. When you look at mRNA, it has codons. And so for this, if we had AUG, we would do the first letter, and then the second, which means we're in this box. And then we would go to the third. And that would tell us right here. So you triangulate, and that would tell us our first codon is the start codon, methionine. I chose AUG because it is actually always the first codon of every gene. And so that's the one codon out of all of these that I ask you to remember. There are other ways to look at amino acids, so I want to show you my favorite way, and that is an amino acid circle. So for this one, we'll use a different codon. I'll say this. You just start in the middle and you work your way out. And that tells you your amino acids. So this would be gly, which I think means, well, darn, I've actually forgotten. But it's an amino acid.
Code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule.
All right, so a couple things here. Again, amino acids aren't just little circles. They have different shapes, just like Lego pieces have different shapes. This is just for simplicity. But we do need to talk about a couple things here. First, this molecule here is actually a piece of RNA called tRNA. And then this here, which also is going to have a big unit later, is called the ribosome. And it is also made of RNA, but it's made of ribosomal RNA. So we've only talked about so far this red piece of RNA here called the messenger RNA, and that was made by transcribing a gene. But there are also ribosomal RNA and tRNA that are made. The 3 of these work together to create a protein. So watch how they work together. Basically, this code right here is called an anticodon. And it will attach to the correct codon. So watch that happen.
The type of amino acid is determined by the anticodon sequence of the transfer RNA.
I do want to add, so basically, these tRNAs here are empty. They don't have these until you eat. once you eat, these amino acids come into your cells, and these tRNA are waiting. Once they find an amino acid, they start looking for a ribosome.
Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site.
So remember that these 2 amino acids connect with what is called a peptide bond. And so as this grows here, you're going to see this is going to be called a polypeptide.
The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached.
And so what has been created here is a long polypeptide. Once again, we have to keep in mind that this is simplifying. These circles are supposed to represent amino acids, but we already know from looking at real amino acids that they have different shapes. Okay, they're not all circles like this. I just want you to be always aware of how complex these 20 different amino acids can be.
Release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation...
So this isn't finished yet. This is just a polypeptide. This polypeptide is not a protein. It has to go through a folding process before it can become that. Once again, we don't know the rules for that. That's why we made that video game Fold It, where you can play a video game to try to determine how proteins might fold. So this needs to be further processed. To do that, we need to put this in a giant vesicle and send it to the Golgi apparatus. So watch as we send this to a Golgi.
The polypeptide may need to be modified before it is ready to function.
There it is. Okay, so here, this is the nucleus. This is the rough endoplasmic reticulum. It is rough because if you look closely, you can see all of these ribosomes. And so inside of this chamber is actually where the ribosomes are making the polypeptides. And so what has to happen here is these little bubbles come off here. You might be able to see them if you look closely. These little bubbles come off, and those are called vesicles. Oops, I spelled that wrong. These vesicles then go to the Golgi where they fuse.
Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive.
So I want to really quick. So that's true for digestion enzymes. So sometimes we do exocytosis, where we will send the protein outside. And so, for example, a cell in the intestine needs to send out enzymes. But sometimes we need to send proteins to things like the mitochondria here to help build it. Or sometimes we need to send it back to the endoplasmic reticulum to build that, or maybe to the nucleus. We've got to send these proteins everywhere. And so these vesicles have to go down cytoskeleton pathways to find their final location. And we've already talked about how they do that. We talked about our motor protein, John, who sits here and he walks along the cytoskeleton, and he finds the appropriate location. But yes, that's how it works. Here in the Golgi, we have that polypeptide, that big long chain of amino acids, and just like a clown folds a balloon animal, you take that polypeptide and you fold it until it finally looks like the final product right here. Well, that's one example. There's lots of examples. Here's another one. Ooh, I like this one. So all proteins have different shapes, and those different shapes determine their function. All right, I hope that all made sense to you. I guess we'll find out when you do your quiz.